And now we can get started. So I'll introduce our speaker today, Dr. Steve Newmaster, Professor and Botanical Director at the Center for Biodiversity Genomics in the Biodiversity Institute of Ontario, and Chief Curator of the OAC Herbarium at the University of Guelph. Dr. Newmaster has been a genomics professor at the University of Guelph for 20 years and is currently the director of the Natural Health Product, or NHP, Research Alliance. He conducts research on natural health products and molecular diagnostic identification systems with more than 150 publications and $20 million in R&D funding. Currently, he is conducting leading international research on molecular diagnostic tools for NHP authentication, genomic diversity in botanical ingredients used in food, probiotics, pathogens, nutritional supplements, medicinal plant, and natural health products. His R&D has resulted in the development of policy on biodiversity and international trade of botanicals, including product authentication certification within the food and natural product industry within Canada, the US, Europe, and Asia. Most recently, Dr. Newmaster is leading the research at UG behind a portable, inexpensive diagnostic test kit for COVID-19, which is being used to confirm coronavirus infection in patients in Europe and to detect the virus on surfaces in the United States. So Steve, please take it away.
I'm actually alumni too. I did my degree in botany genetics at the University of Guelph in the 80s. A wonderful program that just set off my career and love for botany and medicinal plants. Behind me is a trip that I was on last year in the northern Amazon in Brazil, in an area that was been locked down for several decades due to political unrest and drug trade. And it was recently opened up. So a number of botanists and geologists, we went in exploring. And those are big lily pads you can see behind me that can actually hold the weight of probably a cat. So I'm going to stop my video and move into my presentation. I'll turn it on later when we can have a chat. So pleased to meet everyone. So today's discussion is a story. And the storyline I show in a storyboard on the right-hand side. And I'm sharing this from my research program at Guelph, the NHP Research Alliance, where I have a wonderful team who I'll just briefly introduce you to in the background. And some of these people, many of them PhDs, but some of these people have been with me almost 20 years, like Dr. Raghupathy, who you'll see throughout this presentation as we've gone through and made many discoveries together. So the question of the day is about natural health products. And the premise of the work that I've done for my entire life is understanding how plants heal, not just as a medicinal plant, but also in nutrition and as food. And as I've traveled around the world, and I've been on every continent, and I've been in many remote cultures, sometimes living with people for months at a time to understand their language and their culture and how they use plants. And every time we're going in, we're asking a similar question. First of all, to understand the taxonomy, how do you classify, how do you identify these plants? To understand biological variation, and then we collect them and we extract DNA and understand genetic variation and metabolite or phytochemistry of the plant, but then to ask them how they use them. And I've got a background as well as a minor in anthropology. So we learned how to understand the knowledge that people have around how plants have been used as healing. And they've become important in setting hypotheses. In recent times, as I've been doing my work, we've discovered that we need to make sure that plants are effective in healing and that they're pure. So part of the story is about the purity of plants, that they're authentic. Everything starts with a collection. I'm a trained taxonomist, systematist, and we classify plants. We name new plants. Some of the plants that I was collecting last year in the Amazon were new species. They're described in Latin, they're pressed, they're mounted, they're in the herbarium. As curator of the herbarium at the University of Guelph, which has a rich history going back to the original OAC, has many collections, some back from the 1800s. And if you come visit me, you'll be able to see these collections. And you can see here, there's a strawberry plant and a ginkgo plant, both which have medicinal value. So we have about 250,000 collections, all mounted. We have the geographic location. They're all online in the Herbarium Information Management System, which is online, HIMSS, which you can find if you link to my website. We collect these plants and we use these as not only an archive of the taxonomy, but also as a reference material library for doing research. That reference material library, from my perspective, includes both genomics and metabolomics. And I'll be talking about genome scans and fingerprints. I'll also be talking about DNA barcoding, which we worked on almost 20 years ago, and now we've gone to more advanced techniques. And then lastly, I want to talk about how this is an important tool for industry. And one of the reasons that we've reached out to work with industry to make sure that products are authentic, because at the end of the day, what's in the bottle when you buy a product is what you purchase, which is incredibly important to you. But from my perspective, to have efficacy and for that plant to be experienced by you, it has to be the authentic plant. And how do we do that? And we have tools to work with industry to make sure that we have a proper identity and purity from a phytochemistry perspective. So most of you know where we are, and maybe some of you have visited the Biodiversity Institute. We have two buildings, the original Biodiversity Institute, which is on the kind of the bottom left, and then the Center for Biodiversity Genomics. And that front-facing area that you see, the whole upstairs floor, is the herbarium and the research lab that I work out of. It's important to show the griffin, and I often show this slide when I'm doing international presentations and talk about Canadian, where I'm from. But as the guardian of knowledge, knowledge is incredibly important to my research program, because we look at the perspective of knowledge from two different ways. Science, which is the core of what we do, and building science and publishing on science, but also traditional knowledge that we respect when we go in different cultures. It's an ancient knowledge. It has incredible value. And the intersection between that knowledge and scientific knowledge is an opportunity to build new knowledge. And that's been a premise of my research program for 20 years. Here's my wonderful team of people who work with me. It's only some of them in the lab. I often have between 20 and 30 scientists who work for me, multiple graduate students, and it's just a joy to be able to build a good team throughout the years, and then have these people go work and become colleagues in different places on the planet. I also want to recognize the people I've been working with in the remote areas of the world. This is going down the Amazon with local parataxonomists. These are people who have the same love that I do for plants and have grown up to know plants and learn them in their own villages, not just traditional villages, but they may live also in some of the more populated area, like Sao Paulo in Brazil. And I meet up with these people and they bring me safely into areas, and we work and we live closely together. But then there's the people who we meet who don't have much connection between modern society and where they live in localized areas. And it's those people that we spend time with to understand plants, their taxonomy, their classification, how they use them for healing. And then there's a picture of a project that I worked on for quite a while on nutmeg. And those are actually the nutmeg seeds and the arils around them. It's a source of medicine. It's a source of very complicated chemistry. And there's many species, many of which you've never heard of, many psychotropic species that are very bioactive. I've spent more than 20 years working in Asia, particularly India and the Himalayas, Bhutan, Tibet. And these are some of the people who I've worked with in the communities. And some of the markets where we collect and learn just in a marketplace where the plants come from. I want to be able to give you a narrative to understand some of the background and knowledge that we have. In that bottom left corner is Dr. Raghupati, Raghu, like spaghetti sauce, who's my best friend and has worked with me in the lab at Guelph for 20 years. Palasami is an Aboriginal healer who you see there in the white shirt, the white muscle shirt. He lives in that hut. And we would go off collecting in the Nilgiri Biosphere Reserve in the Western Ghats, or the small Himalayas in India. You can see he has bare feet. We all had to be in bare feet because it's a very religious area. And there's many snakes, which makes it interesting wandering around. On the right-hand side, you see plants with tags. You see moss and lichens in that bottom right-hand corner. We collect these and use an anthropology technique called pile sorting, where we put a label on a plant and we ask them, we put the plants out and we ask them, what is this? What do you call it? Where do you collect it? How do you use it? How is this important to your society? And we gather up that knowledge, and then we extract DNA, and we're looking at phytochemistry, and we're trying to understand how these plants heal. What are the mechanisms of these plants? And the people share this wonderful knowledge with us. It's what we have in common when we meet them is, when we know the language, we have that, which is part of their culture, but it's a love for plants and the knowledge of the plants. And we have great respect for one another. Often when I'm out with collectors, it's a very different experience than how I was trained back at Guelph, to have a hand lens and look at flower structure and count petals, look at the position of the ovary and the fruits and placentation to understand the number of carpels, so I can classify a plant scientifically, as in a Linnaean classification. Now when we go through the forest, they're bending the plants, they're touching them, they're smelling them, they taste them. They have different characteristics, but there are similar methodology and having characteristics to be able to identify. And we collect these and understand these. And it's absolutely fascinating, because the plants have structure and they have phytochemistry that gives off odors and smells that we can measure. And they're cues that they use to be able to identify. Even different colors are different pigments in the plants. We've worked quite a bit in China, especially within the tea industry. And believe it or not, today, I don't know if you've had the wonderful experience of traveling to China and seeing how tea is collected, but it's still collected like this. I don't think I have the picture in here with a big sign, Herbalife, one of the companies that I work with that's in Southern California. They have a large tea processing facility in Changsha, which I've visited before, and then gone to the tea collecting to collect tea and do sample preparation and to understand chemistry and genetics of tea. There are many different cultivars of tea, and they have different qualities and different price points. And it's important to have these tools to understand the quality. It's quite a transition to be on a high-speed train, whipping through the countryside at 500 kilometers an hour in China, looking at people with a wooden cart pulling tea leaves, and then get off and go into these hills. It's quite a brilliant thing. Quite love it. This is in Taiwan, collecting millet, proso millet up in the mountains, the Tile Mountains, with the Tile people in the Brunei who have grown this for hundreds of years. Kevin Birch on the left-hand side is one of my PhD students that worked on this project. A picture in Taiwan going through the jungles. In the Himalayas, sometimes there are dangers with the job. As you can see, I'm climbing up to do some work and collect samples that higher up. And working with the industry and teaching about quality of tea and how to collect and how they're graded. And that's me in the picture in the blue coat, in the center of the light blue coat, second from the right. And collecting and working with colleagues around the world. That bottom picture on the left-hand side is in Hong Kong at a botanical garden and herbarium where we share specimens and we share data. And it's a great privilege to work with these experts around the world and do collections and share data and train. And that's what we do as we work together and share knowledge. So we have quite a community. And this premise of the NHP Research Alliance is about an alliance, not only with industry and the University of Guelph, but researchers around the world. And we have a great relationship with the Chinese Institute of Medicine. Some of the farms that you see when you grow, when you're traveling around, are not the characteristic farms you see when you're traveling around Southern Ontario. So to be able to travel around and see chocolate farms, or to see farms that have great acreages of medicinal plants growing with other plants, such as pepper plantations, and then medicinal plants growing beneath. So agroforestry in practice by the locals is such a wonderful experience. And we not only collect plants and we collect mushrooms, but one of the frontiers that we work on is also looking at soft corals, not hard corals that are very fragile, but the soft corals that grow very quickly have complicated chemistry. And we're looking at the phytochemistry to try and understand new phytochemistry that may be important for pharmacy. Actually, right now, I'm down in Mexico collecting plants, and in another week, I'll be out in a coral reef area doing some biosphere work for the reefs, but also collecting some of the soft corals to do phytochemistry on them. And at times, you run into sharks. And sharks actually have a really good place in my research program. Sharks, as people maybe have a misconception, are actually fairly docile and have a very specific diet. So they're not really interesting. That's me photographing the reef shark as it's going by. They're not really interested in divers. They're interested in eating specific types of food. One of the problems that I've learned about in the industry is in both food and cosmetics, you find squalene. And squalene makes up a large portion of the shark body, and it's body fat, body oil. And it's collected because it's has very good healing properties for the skin, and also transmission of different chemistry into the skin. So it's a very good base to use in cosmetics. Shark meat is also very accessible in areas, especially off the west coast of South America. So sharks show up in human food. We've heard about this in food in fish stands, and we've also heard about this in pet food. So some of the canned cat food have found shark in, and then cosmetics for squalene. Why that involves my program is we design tests to be able to identify in natural products the ingredients. And one of the tests that we've designed is to identify shark squalene and shark species using DNA techniques, so we can keep sharks out of our food supply as many species are starting to go extinct. So here's where I get into the science as I'm moving through. And I want to start off by saying that all living things have DNA, and the DNA is a double helix. And when you split it apart, these base pairs can be read like a book, and they can be filed like a book. And that was the premise of DNA barcoding. And if you collect samples and you take a sample from it, an extraction of DNA, you can then use that sample as an archive and a record to go with the DNA itself. Here's an example of a sample that's collected. It's ginkgo biloba. You can see that we archive this into the herbarium. We do the typical Linnaean taxonomy and identify it. So these leaves actually have that wedge-shaped base. They have a dichotomous venation. If we zoom in and look at the veins, all diagnostics that I use of the vegetation to understand that it's ginkgo. And then we extract DNA. And that DNA has a specific sequence that matches in the library of ginkgo. So we have this process, and this is the DNA barcoding process that we go through. That leaf in the top is strawberry. So it's Fragaria vesca. Why I know this is as a botanist, all those teeth go up to the apex of the leaf, that biggest leaflet that's pointing down, and the smallest tooth is smaller than the ones adjacent. And that's in the key to separate Fragaria vesca from Fragaria virginiana, the common strawberry. These are techniques that we use to be able to identify it. But when we extract DNA and we put it into a library of many strawberries, now anybody, you could collect it and send it to a facility, extract it, where they extract DNA out, match it to a library, and they would come back and tell you. And this is a modern way to be able to do identification of plants. And our goal is to not only bring this to society, so to democratize molecular biology, if you will, to bring it to industry, to help them in quality control so we can have better products, but also someday to have the tricorder that you would just be able to pull, so some child could pull it out of their stocking at Christmas and it would be cheap and easy to use, and they could walk around in the backyard and learn plants. The question that we've wrestled with now for almost a decade is what's in the products? The question in my research program and several others. What's in this project the products that we're consuming? And we assume, and I once did, that it's exactly what's on the label, but sometimes it's not. We built a library of medicinal plants in 2010 and 2013. My graduate students and I sampled the natural health product industry, just ordering either online or buying in stores and shelves throughout North America. And we found out that there was a lot of adulteration. It was considerable. A third of what we looked at did not match what was on the library. And this became a very important publication, of which many other publications have now been written, to look at how adulterated these products are, but more importantly, to come up with solutions. So this was published in a medical journal, and down on the bottom, you see this ultrametric score of 860. It's a measure of how much it was cited in the news and picked up by media, how many times it was downloaded, how much it was cited in other scientific literature. And it's in the top 5% of all publications, this article, ever published. Why? Because people were stunned that the products they were buying for their health may not be what they think they are at a level of 30%. And this was published in 2013. This was picked up and made into a number of documentaries, which you can find. So you can go to The Fifth Estate and Frontline. You will see me dancing around on screen talking about the same things that I'm talking about to you today. And that was back in 2013. So a couple documentaries were made as well. It was very much taken up by society. And now these are actually books that have just come out in 2020. Many books have come out, but these are books that we've written, and with my colleagues, we've co-authored several chapters in each of these on food fraud and medicinal plants, the challenges for the industry, and then advanced technologies, that book on the right-hand side for quality control in plant-based medicines. In one of those books, you will see the biggest sampling of marketplaces throughout the world done to date. So over 8,000 samples were collected, and we extracted DNA and matched them to labels. And the authentic samples are in the gray numbers. So you can see in North America, 72% authentic, 28% adulteration. And this is a recent study. You can see in different parts of the world. You can see in places like China, where 80% are authentic and 20% are adulterated. And there may be several hypotheses or explanations for that. One of the ones that we wrote about that I think surprised a lot of people is that when I go into a market, even in Hong Kong, where the population is huge, the markets, there's many medicinal plant markets. They're small, and they're found in particular neighborhoods. And when I walk in that store, the owner of the store looks at me and realizes I am not his normal clientele. I'm not only not from there, but I'm not the normal people that come in. And when the normal people come in, it's normal clientele, they have great conversation, they know each other, there's accountability in that store. That's different than when I walk into a store in a larger city, particularly in North America, or I order online. You're anonymous. They don't know who you are. There isn't an accountability. And I think this may be why in some rural areas of the world, like China and India, we didn't find as many adulterated samples when we sampled. They were smaller markets where there was a close community. There's probably other explanations, and that's not in any way statistical, but it was certainly an observation that seemed pretty obvious. So how do we build these libraries? This is Echinacea, you might recognize, and there are multiple species of Echinacea that are used medicinally, and they're used several ways medicinally, sometimes in boosting immunity is one way to be able to deal with common cold. Here's 3 common species of Echinacea. Pallida, Echinacea angustifolia, and purpurea. And this classification, this tree that you see here, is a classification based on DNA. So multiple samples classifying all together into one grouping, which we call a clade, to give a nice classification. The data that we originally had for this, just downloaded from public databases like NCBI GenBank, wasn't a clean classification. We had to go into the genome, sequence large sections to find clean areas in the genome, markers that could differentiate these species in the marketplace. So how do you do that? Well, you collect many samples, and they're archived in herbarium. So every one of these, we have a professional ID morphologically by a taxonomic affidavit, and then we extract DNA and phytochemistry. And then we collect across the range of the species around the world, including areas where it's grown and areas where it is found naturally. So both from the industry supply chain and natural populations. And then we bring in mathematics. And I have a second degree in spatial mathematics. And the mathematics works like this. Sequences from one population, when you sequence, compared to another population, isn't exactly the same. It's like us, human beings. We have a distinct sequence. I have 5 siblings, and my sequence to my 2 brothers is not exactly the same. There are differences. So even within a population, there are small differences, and we call these single nucleotide polymorphisms. One section of the genetic code in one position that would be different when you compare another individual in the population. So we collect lots of populations, and we have variants. And this diagram, which is called an ordination, is an ordination of variants in sequence. I could show it as a tree, mathematically display it, or it is an ordination. And I love these ordinations because they can be in 2 dimensions or 3 dimensions. There's actually an X and a Y and a Z coming out at you. And I see these as clouds of variants, and these are each of the species. A species is associated with an individual that was collected. So if I extract DNA from a product, and you see that pill matches down into a cloud of Echinacea angustifolia, and there's a reference sample in there. And the distance between the 2 can be measured based on sequence difference. So it's very logical and it's easy to read. For the industry, you now have a tool that if you have a sample you're collecting from the supply chain, and it may be an unknown, you're buying a big container of Echinacea from China, and you want to know, is it really Echinacea? Well, you can extract DNA from a sample from it and then match it to the library, and it should statistically fall within this cloud with certain confidence intervals. If it doesn't, it's non-compliant, you reject the load. And that's how we build regulation protocol based on statistics using genetic tools. And we're doing this over and over again. And in my program, we focus quite a bit on doing this and building these tools for the industry. And we're not alone. We have many colleagues that work with us around the world. We do this with collections that come in that we've collected ourselves from botanical gardens, companies that send us their supply chain. We have regulatory agencies that send them. And then we build a library not only for Echinacea, but many species from many collections. So you have this cloud-based mathematical system that's very powerful and has quite a bit of value. And it's not only the genetics that's associated with these clouds, but the phytochemistry, which I'm going to get to in a moment. So we collect the genome sequences, and we're collecting, which I'll talk about in a minute, NMR spectra to have a phytochemistry profile. I realize I'm throwing a lot of stuff at you. We'll have time to chat about this in a bit. This is Adam Fowler down the right-hand side. So he's a PhD student. It's actually just finishing. He's going to be defending just before Christmas. And his question was about DNA. What happens to DNA when you move from a fresh sample, and this is tea, and he was sponsored by Herbalife. That's why Herbalife's down there as a graduate student, which is fantastic. And he went to their site in Changsha in China and then collected in the hills. The tea is processed into a powder, which is an extract powder, and then made into an extract capsule that they sell to consumers. And the consumer, us, we want to know that the green tea's there and the green tea has a health benefit. And the question we have is, well, how well does the DNA tool work? How well validated is it? What happens to the DNA? And it's very mechanistic. As we dive into the science, we want to understand what happens to the structure of DNA as it's processed and it moves through. How reliable will this tool be when an extract powder is made from a sample? And this is very important for the industry to understand. It's very detailed work, it takes a long time. It's the subject of the PhD thesis. There's my great friend Raghu in a cranberry patch. And Raghu led a paper that was just published a year or so ago. And what we found out in that paper from our team is, as you process, and this is an ordination looking at metrics of DNA quality, the fragments become shorter. Well, that's no surprise. DNA is broken into smallish fragments, and there's less of it. But at the end of the day, even in highly processed ethanol extracts, there's still DNA. And at that time, the industry said there's no way industry would be found in these products. And we found considerable DNA, but just in small fragments. So we started to build tools that use small fragments or probes to be able to identify products. It's the type of mechanistic work which is very important. Validation. If we look at other sectors that impact our society, like when we go out and we buy produce and we want to know there's no foodborne pathogens on it, or even if we're buying meat and we want to know there's no E. coli in it, how are these tools put into place and how do we know they work? If you're detecting for E. coli in meat for quality assurance, and you have to do that by law, there's a validated process using a DNA tool. And it's a polymerase chain reaction tool, PCR. And you must go through these steps that you see in this table. And this is from a publication that we worked on as a validated method. So for working with foodborne pathogens, first they build a reference material standard, and then they look at specificity, that you're identifying E. coli, the appropriate strain of E. coli, and not some other closely related strain. Sensitivity, how sensitive is the test? Can you detect small amounts of E. coli so that it is no longer toxic or risk to society? And then the last three, reproducibility and repeatability, is the ability for someone to run this test on different users, running it on that machine at different times in different places. And that's really a confidence statistical metric to know that if you're using it over and over again, it's always going to work. And then the practicality, it's kind of hard to say. Well, how expensive is this test? Is it realistic we could even use this in society? And then we went through the literature and we saw that, well, this is done well for foodborne pathogens, all these check marks. And these are actually referenced literature and standards. And then we looked at meat and seafood to be checked for the species identity. You know, someone like Bob Hanner at the University of Guelph looking at the SushiGate scandal, where sushi is substituted or adulterated quite often. So do they have validated methods? Well, they haven't done as many of these checks for a validated method. Even allergens to check for peanuts in food. Probiotics was missing these steps for validation, and botanicals had the most gaps. So we felt it was very important to publish this paper on this is the process that you follow to have appropriate tools, and then a big guideline working with Health Canada and FDA, the EU, US Pharmacopoeia, AOAC to build out standards that could be used. And we did this then for each botanical. So ginkgo biloba would follow that methodology, and we would publish what is an appropriate test, what is specificity, what is sensitivity. And we did this for probiotics. There's Hanan Shahada, a brilliant scientist in my lab who does a lot of this work on probiotics, and a validated method for probiotics. And probiotics validation for specific strains, not just looking at species of probiotics, but individual strain identity matched to the label. And the reason this is important is because we're finding out more and more that strains have importance to our health. And I'll talk about that in just a moment. We did the largest study to date on a market survey of probiotics going to the shelf. And what we found is actually the probiotics market is pretty good compared to the botanical market or the fish and seafood market. It's not as adulterated, but there is a problem in identifying strains. So in this table that you see here, there's in the first column are a bunch of strains. They're just numbers because that's how they identify the taxonomy of the strain, and they would be labeled on a bottle saying this is in the product, and then a whole pile of products you can see going along the y-axis. And we had many different products. So this is just a segment of it. And if it's green, it's like, oh, it matched to species level. And if it's red, it failed, and it failed at strain level. Or we found a strain which wasn't declared on the label. And sometimes these are cheap strains that they use as fillers. So there's a problem in the marketplace, but it's not as big as we thought, or could have predicted anyway, which is good to hear. There is a definite need for tools, and those are the validated methods that I just showed you. So how is this used in the industry? Well, we build libraries, and we work with suppliers and build libraries, as I've shared with you today. We build tools as a small PCR instruments like the one that you see in the top left. That blue box is a high-risk B cube. And we validate, we make, test, and validate test on it so that the industry can test cheaply and test every batch. It shouldn't be an impediment to have an expensive test or send it to a lab. You can just test on site. You can test anywhere at the manufacturer, in the field, distributors, because at any one of these areas is where adulteration can take place, especially economically adulterated products. Inspectors could use these tools and be able to check products as quick audits. Consumers could connect with labels, especially smart labels, to understand, first of all, it is authentic and it has been tested. It's not just a paper-driven certification program that promises to be authentic, which is all and good, but I'm a scientist. I like to have a test. And I would say that everybody should demand to see some evidence that what they're buying is authentic, because we're buying it for our health and well-being. So the question then becomes, what are in our products? And there are a number of molecular tools that we use to figure this out. And I've talked about genome approaches and genome scanning, but I haven't talked about metabolite fingerprints. So the genome scan, we can identify locations in the genome and how much variance there is. Fantastic. It's a great tool. These are genome iris scans just from cannabis. And you can see in these white circles that, well, some strains are different than others. These are all CBD strains. And you can right away look at it and say, yeah, the genetic variability is not as great. And there's a definite pattern there associated with CBD versus THC-based strains. Another tool that we can use, which has been an innovation that's come out of Guelph in our lab working with Bruker in Germany, is using nuclear magnetic resonance. And in this, it's a non-targeted approach in chemistry where we can look at a broad spectrum of metabolites. And we do this in a big magnet. And the magnet sends through a pulse, and that pulse breaks apart bronze and protons for a fraction of a second. When they come back together, we now model that organism's phytochemistry. Some of the phytochemistry is in there, actually, a good capture of the spectra. So here's golden seal, Hydrastis canadensis, and the structure for berberine, which is thought to be one of the bioactive molecules. And we can see in here that golden seal has a spectra for berberine, but golden seal itself has a more complicated spectra. And that's this blue spectra that you see here with lots of peaks. And those peaks represent different phytochemicals. We can use a fun image like the one below of a fingerprint and translate that mathematically into a fingerprint to identify all the different components that are found in that plant spectra. And that's me standing beside an NMR. So we did this for coffee, and I'm just going to quickly give you an example of coffee. I've been working on coffee for 10 years. And the question is, Arabica and Robusta, two traded commodities, 100% more they get adulterated. But then coffee that's found in different parts of the world, like an Ethiopian Yirgacheffe, versus a Robusta just out of Brazil that has a lower price point, can be huge differences in cost. And how do you identify that? So here's the spectra from coffee. And this is what we're doing for natural health products, but this is a good example because we've already done this. There's a red spectra and a blue, Arabica and Robusta. And we can pick out on those peaks, it's not as if one peak is caffeine, it's actually a pattern of peaks. So you see caffeine showing up in a number of places. So the first thing is like, well, the caffeine spectra shows up in here in peaks and then over here in peaks. And you can see there's a difference in red versus green. So the Robusta has this higher caffeine. It makes it a little more bitter. We can see big differences in 3-CQA compared to 5-CQA, where the Arabica has higher peaks. This is a good identification tool, and it tells us about the phytochemistry in the plant. It tells us about coffee flavors. So the cuppers that are tasting coffee can taste these different notes, and this chemistry can explain it. And this is some of the exciting work we're doing in coffee. So we can tell apart now coffee from different parts of the world. We can tell apart coffee quality. We can classify in the ordination that I was talking about. These are just all different coffee samples breaking into different groups that represent different parts of the world. I can tell now in Brazil and this is a project that we actually worked on. A coffee came back of a lower grade, and we had an NMR profile for it, a metabolite profile, and we found bitter notes, so certain bitter compounds that were found in it. That coffee was attacked by a fungus, and the plant responded by producing these bitter compounds and made a lower grade. We could see that in the fingerprint. So now you have a tool that could be used for grading coffee. This is the same tool that we use in wine for identifying wine varieties and wines of different grades. We use it in honey to be able to identify pure honey. We use it in olive oil, and we build these models. And this will be an incredibly important tool for natural health products like turmeric, which has many different strains. And you can see these fingerprints on the right-hand side of turmeric. There are many different metabolites that are found in turmeric, the same species, but different cultivars grown in different places of the world. And they definitely have different value and price points as a commodity. So we work with the Cumin Association, the global curcumin, to be able to get samples from all over the world. I go and collect them too with my colleagues, and we build libraries. We do this for cannabis and building a cannabis registry of cultivars so that we know that the cannabis cultivar that's being sold is not only the one they say it is, but also next time you buy it as a supplier, and we work with Shoppers Drug Mart to do this, it's the same batch to batch to batch, and that's based on a metabolite fingerprint. And these are metabolite fingerprints from cannabis, and we can identify hemp from CBD strains. We can tell if it's leaf, we can tell if it's bud material. It's not surprising because the plant produces different phytochemistry from different plant parts. The plant responds to different soils and different chemistry to produce different phytochemistry. And in cannabis, it's very influenced by light. And different light wavelengths of light end up producing different phytochemistry and different fingerprints. And we are modeling all of this. So different strains of cannabis would have different metabolite fingerprints. These metabolite fingerprints would classify into different groups. And we can see this in 3 different strains from different companies that broke out nicely into different groups, the green, the red, and the blue. But when we collected from the marketplace, all of these numbers should have clustered nicely together, and the 1s aren't with the 2s, and the 5s are spread around with the 3s, and they should be in nice tight clusters. That's a problem for the industry. That's a problem for users that expect the same experience because there's high degree of variability, and that shouldn't happen. And there's several reasons why it happens in the industry. And we're trying to build tools to make sure that that doesn't happen, and there's regulation. Here's an example of batch to batch variability in cannabis products that we collected and brought in. And it should be one tight circle of clusters, not a classification into different groups. If you're using cannabis and you have this degree of variability in the phytochemistry, you're going to have different experiences, and that's not acceptable. You want to have the same experience every time. And certainly a company like Shoppers Drug Mart does not want to have their users having different experiences. It's the #1 complaint in the cannabis industry with quality, is that they don't have the same experience every time. And part of it is because the supply is actually different. And now we can measure it. And these are just examples that you can, in the cannabis industry, achieve great quality assurance and consistency. 3 different strains, 95% confidence intervals in the metabolite structure. And we've worked with a few companies to help them tighten up their growing protocols so that they have very tight products. And they become the products then that can go to retailers that have quality brands. And then that is for consumers. Set of fingerprints, the blue ones down on the bottom left, is consistency from batch to batch to batch to batch. And the ones that I had up just before it are just different strains. And that represents variability in the metabolites. And we can, just like the coffee, dig down deeply to see which cannabinoids are there and which ones are bioactive. So I'm coming to the end right now. This, as I've already talked about, honey is a good example where it's been used. Milk, we actually run these samples without any prep. We just put them in the NMR machine, and we have distinct profiles for raw versus pasteurized milk, or ultra filtered milk from different areas of the world. We've looked at probiotics, and we can see that different strains of probiotics produce different metabolites. And it's those metabolites that are bioactive in the body. And it's the same story that I've been trying to share all through this presentation. These metabolite profiles, whether they're from probiotics, or you're taking them in nutrients like a glass of milk, or you're taking a natural health product, are what interacts with your body to give you a healthy lifestyle. And that's the way it's been for many years in these rural cultures that we spend time with. And in modern science, as we look at clinical trials and understand how these different products are interacting with the body, we're seeing great associations. And these are some key articles that are coming out, and understanding that, well, at a probiotic level, it's incredibly important to understand strain, because different strains have different impacts on the body. And these are just a few examples I wanted to share with you. This is the modern era right now of natural health products. Clinical trials, understanding bioactivity of molecules, collecting libraries from them, and the clinical trials looking at inputs into the human body with NMR profiles. And then we also look at outputs in blood and urine and human health markers to understand what these actual metabolites from plants or from probiotics are doing in the human body. And this is where I end up at the end. It's not just an identity tool to make sure things are healthy, but now it is a tool to understand how plants and probiotics, different foods, actually heal our bodies over time. And this is changing in society. And it's the clinical trials intersecting with a lot of other sectors that we never thought would before. So taxonomy, molecular biology, phytochemist, looking at chemistry, all working together to understand these patterns. So very exciting times and big teams working on this. I go back to some of the wise people I've worked with over time that at the end of the day, talk to me about this knowledge of how things heal and what you put in your body. And it's interesting in First Nations, especially in the Anishinaabe, and I studied in Anishinaabe with Basil Johnston at the Royal Ontario Museum many years ago, back in the 70s, late 70s. And big words like Indo-Kagedwe are about knowledge of plants and animals and how that intersects with us and helps us to live our lives and live a happy life, improving life, one of the things we want to do at Guelph. But when I go and work on Ayurveda in India, there are terms such as the Rishi and the Siddha that talk about the same concept. As humans, we're coming up with the same concept. And today in modern science, as we're looking at collectively these plants we take in their body, how do they heal? These same concepts are coming back again. And I just find that fascinating, how this ancient knowledge and new knowledge are intersecting still now. So I'm going to stop now and open up for questions. And you can contact me anytime.
Yeah, that was really interesting. I love listening to this talk. So thank you for that. We do have a few questions in the chat. One that's kind of coming up a few times is kind of where would a consumer go to for information about just being more educated and making decisions on before purchasing natural health products, and how do they kind of guide their decision making with making purchases and knowing that they're buying something that's actually effective?
I think it's wonderful that like the people in the audience right now are interacting with the university, and the universities are the great source of knowledge. In the early days, we would say, oh, don't go to the internet, but universities are on the internet. So it depends. You go to those sources on the internet where you can find great knowledge. And those are in the scientific publications, universities, great museums, like Smithsonian Institute, who we work with. Those are places, hospitals, clinical hospitals. We work with Johns Hopkins. I work quite a bit with UCLA. These are areas where you have credible information that's based on science and science facts. So you want to be careful where you're getting it from, and those are great places to go. And we're publishing, we publish in scientific literature, but we're sharing more as scientists in social media. So I share quite a bit on LinkedIn as trade articles to help educate everybody on how the science, translating the science that we're publishing into how it impacts us in everyday life. So I encourage you to go and follow some of those scientists that you like, and then look at their networks and then connect to other scientists as well to get right kind of right from the horse's mouth some of this knowledge and understanding of the knowledge.
And with respect to the NHP website that you have, a lot of the work right now is focused around industry. Are there plans to put resources for consumers and individuals to be able to better guide their purchases and decision making?
Yes, our NHP Research Alliance is starting to mature now. It's only a few years old. We built up a core of science to work with the industry, and we've engaged industry to work with them to give them tools. And now we want to start translating this core of knowledge and sharing it with the public and consumers, and also asking what types of things are important to you, because they could become research questions, and then we could share back. So I would say stay tuned to some of the new innovations that are going to come out on both our social media and Natural Health Product Research Alliance website. So it's going to be renovated for the new year, and there'll be more information that's coming out for you.
Great, thanks, Steve. And do you have, can you maybe start, I'm just reading this question. Are there any collaborations that are happening with Indigenous people in North America specifically?
Oh, there's many, with many universities. And there's interesting questions that are coming out. So in Northern BC, we're working on a plant called squaw root. And this is just our example that I can share. And I have many like this, but squaw root is used in, first of all, there's different populations of which some are rare and there's some that are common. So there's a taxonomy question, and they're very closely related and they're difficult to tell apart. The healers use them in different ways, and they recognize they're also their value to the ecosystem, and they can identify them. So we've been working with healers in North BC to find these populations, understand how they use them for healing, and it's incredibly important work, but it builds on this cutting edge science that we've been doing.
Yeah, that's great. Melissa had a great suggestion for a future webinar, so we'll pass that on to you so you can maybe share that at another time. Wendy is asking, which cannabis companies are you working with right now, based kind of focused on that consistency?
So we've set up a registry in Canada for cannabis that they can register their cultivars, and we create genomic metabolite fingerprints, and then any retailers that are working with these companies would be able to use these tools to make sure that they have the right strains. And at some point, we need to make this public and let the public know and interact with the public. I'm not sure how to do that. And I'm not sure if I'm even at liberty to say who's doing this and who's not doing this. So I would say, sorry, I can't really divulge that right now, but we are going to be putting this out to society so that consumers will be able to see this. And I think some of the mainstream brands like Shoppers Drug Mart are going to be marketing this out so you can see. And it just fascinates me that they want to engage in scientific tools that are validated to make sure that the consumers are getting a good product. They really care. And that's just fascinating to me to see industry stepping up and doing that. Real leaders.
Absolutely. Joanne is asking, is the percentage of adulteration of products in North America still around 30%?
Yes, unfortunately, it's still around 30%. Part of it's a problem right now and a change in the industry from raw material supply to extract powders. And those extract powders are being mixed up. They're just white powders. So part of this is a problem that the industry is having in just authenticating the powders. So we had to make new tools, and the NMR metabolites work really well, but it's caused a new problem in the industry, and we're working to get these tools in place. The reason they do that is it's easy, it's more stable to ship. It's easier to ship a powder rather than a bunch of nuts and leaves, dried leaves, and they don't have as many fungal contaminants, but it impacts the quality of the material, and the regulatory system hasn't kept up with a way to be able to test. So in the end of the day, to answer your question, adulteration is still a problem.
Okay. Thank you. That's still troubling to hear for sure. Matt is asking, are there any technology companies, Google, Amazon, etc., helping with the management of big data and classification?
That's an interesting question. I'm not sure I fully understand it. So companies that do like certification programs, or I probably won't have time to respond. There are a lot of companies who are what I would call stakeholders in the supply chain. And some of these are trade associations linked with companies, and some of them are companies that specialize in supply chain management. And there's many of them around, and they are working with scientific tools. Some of them are, some of them are not. And I think the ones that are not are going to disappear because they must be based on science. And I think they'll just be removed from the system.
Right. And I guess another question just that's a bit more timely is, I know a lot of people are thinking about immunity right now because of COVID. Is there a natural health product you would recommend, or a specific ingredient or botanical that helps to boost immunity?
Yeah, so there are a number of them, but one that has a claim is elderberry, and it's been a hot commodity in the marketplace for immunity boosting. And of course, with supply and demand, when there's demand goes up multiple fold, then adulteration goes up as well because people are adulterating. So now elderberry juice and elderberry has been contaminated with things that are easier to get, like grape juice. So now I need to make a test, and we're working with the Elderberry Association to be able to test and say, yes, elderberry, no grape juice, or vice versa, or if there's a mix. And those are challenging questions. So you can see it's very dynamic, and it's based on the market. And as things become more expensive, people figure out how to make a buck off of it. And we're there to keep things authentic. Mm-hmm. So it's related. In COVID-19 has created a number of problems for the NHP industry, and elderberry is one of them. But there's a lot of claims that it is a very good product, and some of these are clinically based research that's coming out.
That's great. That's really interesting. I know a few were asking about the website. I think Jackie just added that in. I don't know if someone was asking about the names of your books. I don't know if that was a slide originally where there were some, I think maybe the food fraud books, I think they're asking about.
Yes, I can make that available on a link to everyone. We'll be putting it on the website, but we could probably share that somehow with everyone after this.
I don't know if you want to pop it in the chat now, or if that's something we can do later. We can follow up at a later time.
Sure, I'll put that, and I'll put my LinkedIn site where you can also find a lot of this information. Okay,
great, thank you. So I think that's all the time we have for questions. It's 1 o'clock right now. Thank you to everyone who submitted questions. If you have any additional questions, Steve's email's on that slide there. I'm sure he's more than welcome to chat again. And you'll see we've also put in the chat, we have other webinars that we are scheduling in the winter semester in 2021. So please keep an eye on your inboxes for invites or register from our website. Thank you again to Dr. Steve Newmaster for this really informative lunch hour. Thank you to everyone for joining, and hope you all have a wonderful day.
Thank you everyone for your time. My pleasure.